Hi, welcome to Chapter 8, Microbial Metabolism. This chapter covers how microbes capture, store, and use energy. We'll focus on sections 8.1 and 8.2 today, which set the foundation for understanding microbial energy flow. Here is the Chapter 8 outline. Again, we're going to cover these first two sections in this video. By the end of this section, you'll be able to describe metabolism, autotrophs and heterotrophs, energy and electron carriers like ATP, FAD, NAD, etc., enzymes, and enzyme inhibitors. Metabolism is the total of all of the chemical reactions in a cell, and they're composed of, or metabolism is made up of, both catabolic and anabolic reactions. In catabolism, you're breaking molecules down and releasing energy, and these are known as exergonic reactions when energy comes out. Anabolic reactions, or anabolism, builds molecules and requires an input of energy. So these are endergonic reactions when you have to put in energy. So cells are constantly balancing catabolism and anabolism to stay alive and grow. And we can see these reactions down here as well. In anabolism, you're going to add in energy to build larger molecules, whereas in catabolism, you're going to take larger molecules and break the chemical bonds to allow the molecules to be separated into smaller components, and this will allow you to release energy. So energy is going to come out of the reaction. Organisms can be classified by their carbon and energy sources. For carbon sources, we can either have autotrophs or heterotrophs. Autotrophs use carbon dioxide. They're able to fix inorganic carbon into organic forms. So examples of organisms that are autotrophs include plants and cyanobacteria. Heterotrophs are like humans in that we have to get our carbon source from another organic compound. So humans, E. coli, we are unable to fix carbon. So we can't turn something like carbon dioxide into glucose. We have to get our carbon from another source. And the other source is usually an autotroph. In terms of energy down here, energy sources can be subdivided as well. We have phototrophs that get energy from light, so these are photosynthetic organisms, or chemotrophs like humans that get our energy from chemical bonds. Chemotrophs can be further subdivided into organotrophs and lithotrophs. Organic trophs get their energy from organic compounds, so humans and fungi are examples of organotrophs. Lithotrophs get their energy from inorganic compounds like hydrogen sulfide or iron. And this is unique to microbes. So humans and other animals are not lithotrophs. And our book provides a nice table that combines the two, energy and carbon sources, to create categories that we see here, these four. So remember, chemo and phototrophs describe where the energy comes from. If you are a chemoautotroph, for example, you get energy from chemical bonds and you are able to fix carbon. Here, if I'm looking at, I'll choose this one as an example, this is energy from light, and these organisms are not able to fix carbon, so they need to get their organic carbon source from another organism. So our first checkpoint asks, what's the difference between catabolism and anabolism? Catabolism is when we break down complex molecules. That's going to release energy. So these are exergonic reactions. Anabolism is when you build complex molecules from simpler ones, and this requires an input of energy, so anabolism is endergonic. And cells continually balance both processes to maintain energy and structural needs. For the second one, explain the difference between autotrophs and heterotrophs. Autotrophs use inorganic carbon, like CO2, as their carbon source, and these include plants or cyanobacteria, whereas heterotrophs require organic carbon compounds like humans and most bacteria. Energy in metabolism is transferred by electrons. Oxidation means a loss of electrons, and I can see that here. Here we're losing electrons. Reduction is a gain of electrons, and I can see that in this part. Redox reactions always occur in pairs, and redox, the term redox, comes from the combination of these two words, reduction and oxidation. This allows energy to be transferred in small, controlled steps. Shortcuts to remembering redox reactions and what's happening for the electrons include either this one, Leo the lion says Ger, where lose electrons is oxidation, and Ger stands for gain electrons is reduction. Or other courses sometimes use oil rig. Oil meaning oxidation is lose electrons, reduction is gain electrons. Energy from nutrients can be stored in high energy molecules. These include electron carriers like NAD, FAD, NADP. So these are the oxidized versions with fewer electrons, and these are the reduced versions with more electrons. They've gained electrons. ATP is the energy currency of the cell, and these ATP molecules store energy in their phosphate bonds. Breaking these bonds can release energy to power anabolic reactions, while phosphorylation can regenerate ATP from ADP. So ATP can usually be broken down in this format, where one phosphate group is released, and that is called inorganic phosphate. Sometimes, it's not as common, ATP can be broken down further, producing adenosine monophosphate and 2-phosphate molecules, and this is usually called pyrophosphate. So breaking down ATP into ADP plus inorganic phosphate is exergonic. Energy will be released, and that can power other reactions, whereas building ATP from ADP plus inorganic phosphate requires an input of energy. Remember that metabolism is composed of a combination of catabolic and anabolic reactions. In other words, they're composed of exergonic and endergonic reactions in combination. So I'm going to start here on the left. I have glucose, and if I break down the chemical bonds in glucose, I'm going to get energy. So breaking down the bonds in glucose will release energy, and that's exergonic, and I can use that released energy to build ATP. So I'm going to use that energy to put adenosine diphosphate together with that inorganic phosphate to build my ATP molecule. Later on, if I want to perform endergonic reactions that require an input of energy, I can, so this is an example of an endergonic reaction. I'm taking a simpler molecule like glucose and forming a more complex molecule like a polysaccharide. This is endergonic. So it requires an input of energy. I can get energy from ATP that I stored or produced earlier. So if I break down ATP into ADP and inorganic phosphate, that will release energy to power this endergonic reaction. So these are coupled together so that a favorable reaction can be coupled with an unfavorable reaction to make them happen. Right, so electron carriers like NAD, FAD, NADP, these shuttle high energy electrons. So they store and transfer energy in controlled steps for metabolic pathways that we'll see soon, and these can be either catabolic or anabolic pathways. Enzymes are biological catalysts. They speed up reactions by lowering activation energy. And they lower activation energy by binding to the reactant molecules and holding them in such a way that the reaction will speed up. So in these reactions, a substrate will bind to the active site of the enzyme. And enzymes will often change shape slightly for the best fit of the substrate. That's called induced fit. So here I can see an enzymatic reaction down here. The substrate's going to bind to the enzyme at the active site. The enzyme substrate complex forms once that happens. The substrate's converted to some kind of products, and then the products will leave. At the end of the reaction, the enzyme is unchanged so that it can continue to catalyze the reaction. Overall, enzymes are optimized to work best under their typical environmental conditions. For example, microbes that live in hot springs are going to have enzymes that work best at high temperatures, whereas microbes that live in human bodies are going to have enzymes that work best at human body temperature. So let's look at this one, for example. Looks like enzyme activity is best at that temperature, and if this is a microbe that lives in human bodies, that would be 37. If it gets too hot, the enzyme activity will decrease, and the enzyme could even denature and lose its function. If it gets too cold, then you see the same thing. Enzyme activity goes down, and if it's too cold, the enzyme activity will stop. Similarly, the same thing is true for pH. So enzymes, most of them will work best at neutral pH, but microbes that grow really well in acidic environments, for instance, are going to make enzymes that are optimized to work best at low pH conditions. For substrate concentration, enzyme activity can be increased at higher concentrations of substrate. So basically, if you have more substrate, then you're going to have more of the reaction, until at which point you reach a saturation point. This is the point at which you no longer have any more enzymes available. So there's no more enzyme available, or no more active sites available to bind additional substrate. Enzymes function by lowering the activation energy of a chemical reaction inside the cell. And the activation energy is the energy that you need to form or break chemical bonds and convert the reactants to products. So in this graph, they're showing you the reaction without the enzyme or catalyst in green. So this is the activation energy without the catalyst. And here's the activation energy with the catalyst. So activation energy in this case, without the catalyst, you can see is much higher. With the catalyst is much lower. It turns out that many enzymes do not work optimally or even at all unless they are bound to other specific non-protein helper molecules. So these helpers can be cofactors or coenzymes. Cofactors are inorganic ions like iron, magnesium, or zinc. Coenzymes are organic molecules, often derived from vitamins that are required for enzyme action. If we look down here, an apoenzyme is the inactive form of the enzyme that does not have its required helper. So here's an apoenzyme down here. A holoenzyme, it's like sounds kind of like whole or entire, is the active version of the enzyme that has the helper bound. And I didn't see this in the book, so I wanted to make a quick comment that not all enzymes are proteins. Most of them are. Remember, proteins are made of amino acid subunits, but there are some enzymes that are just RNA molecules. RNA molecules that act as enzymes are called ribozymes. So one example is in ribosomal RNA. So ribosomal RNA acts as an enzyme by catalyzing the formation of peptide bonds when we're making proteins. So all ribozymes, these RNA molecules, are enzymes, but not all enzymes are proteins because some of them are ribozymes. All right, so what role do enzymes play in chemical reactions? They are biological catalysts that lower activation energy. They speed up reactions without being consumed. Remember, at the end of the reaction, they're the same, so they can continue to catalyze that same reaction. And enzyme specificity ensures that the correct substrates bind to the active site, and you form the products efficiently. So not anything can just bind. Usually enzymes are very specific for the substrate that they will bind to. Cells can regulate enzymes using inhibitors or activators. Competitive inhibitors block the active site. One example are sulfa drugs, which are used to treat bacterial infections because these drugs bind to the active site of an enzyme within the bacterial folic acid synthesis pathway. So in bacteria, they need folic acid to make DNA and RNA and proteins. In humans, we are unaffected by these drugs because we get our folic acid from our diets. For non-competitive inhibitors, these are also sometimes called allosteric inhibitors, the inhibitor binds somewhere else, not at the active site, somewhere else, usually called the allosteric site, and this results in the shape change of the enzyme. So I'll show you in a later picture, the active site will change shape so the substrate can no longer bind. We also have allosteric activators, and these are molecules that will increase the activity of the enzyme. And what you're going to see here soon in a picture as well is the activator will bind to an allosteric site that optimizes the active site of the enzyme so the substrate can easily bind. Finally, we had feedback inhibition. This is when the end product of a pathway is going to shut the pathway down earlier in the pathway. It'll block an enzyme in the early or beginning of the pathway, saving energy. Here's a visual depiction of those inhibitors. On the left, we have a competitive inhibitor, which binds to the active site and prevents the substrate from binding. So a competitive inhibitor is a molecule that looks similar enough in shape to a substrate that it can compete with it for binding to the active site. It's kind of like playing musical chairs, where there's only one chair, but maybe two people trying to sit in that chair. For a competitive inhibitor to be effective, the inhibitor concentration has to be pretty similar, pretty equal to this substrate concentration. For a non-competitive inhibition, the inhibitor does not look like the substrate, and it's going to bind to a different site, not the active site, but instead an allosteric site. Binding to that allosteric site will change the shape of the active site so that the substrate can no longer bind to the active site. All right, earlier I showed you non-competitive inhibition, which is the same as the one shown here, allosteric inhibition. The inhibitor binds to the allosteric site, changing the shape of the active site so that the substrate can no longer bind. But we also have allosteric activation. In the activator over here, we can see it's going to bind to the allosteric site. This time what the activator does is enhances the active site. So the active site is optimized for the substrate to bind and for the reaction to happen. We also have feedback inhibition at the bottom shown here. Sometimes this is also called end product inhibition, end product, because this is a nice way for metabolic pathways to regulate themselves. So let's look at the pathway to start with over here. So it looks like some substrate is binding to enzyme 1, and that enzyme 1 is going to turn the green substrate into this purple intermediate. The purple intermediate binds to enzyme 2. Enzyme 2 turns A into B. B binds to enzyme 3 and is converted to my final end product over here. But let's say this pathway is going along and now I have way too much end product. I want to stop this pathway. How do I do that? Because I don't have light switch, like a switch and on-off switch in my cells. So what I can do is feedback inhibition. I can make sure that the end product of this pathway is actually an allosteric inhibitor of one of the early enzymes in the pathway, in this case enzyme 1. So when I can see here that when I make too much of the end product, it binds to the allosteric side of the enzyme, changing the shape of the active site, and now the original substrate can no longer bind. And over time, this pathway stops and I no longer generate end product. When the end product runs out, this will fall off of the allosteric site, allowing the enzyme to resume its normal process. All right, so we just saw that competitive inhibitors mimic the substrate. They're going to have a shape similar to the substrate, and they are going to bind directly to the enzyme's active site. So this blocks substrate binding, for instance, in those sulfa drugs I just described. Versus non-competitive inhibitors, they bind to a site other than the active site, and we usually call that the allosteric site. This is going to causes a change in the shape of the active site that reduces enzyme activity, and it requires lower concentrations. So non-competitive inhibitors, you don't have to have as much because they bind to a different site. They're not competing against the substrate. All right, let's recap 8.1. Metabolism is all of the chemical reactions in a cell. This includes catabolism, when we break down molecules and release energy, versus anabolism, when we're building molecules, and this requires an input of energy. So metabolism really involves both and maintains the cell's energy and structural balance. For autotrophs and heterotrophs, remember that autotrophs use inorganic carbon dioxide as their carbon source. They can fix carbon, turning it into something organic like glucose. So plants and cyanobacteria are autotrophs. Heterotrophs use organic compounds for their carbon source, such as humans or bacteria like E. coli. So they're different in their source of carbon, whether they're self-made carbon fixation organisms or whether the carbon is consumed, like by humans. They're similar in that they both rely on energy pathways to build and use molecules. For redox reactions, the third bullet point here, they involve electron transfer between molecules. Remember that oxidation is losing electrons, reduction is gaining electrons. So redox reactions allow for controlled release and transfer of energy, which is going to ultimately fuel ATP production. For the next one, ATP, FAD, etc. So what are those molecules? ATP is the energy currency of the cell. It's going to store and provide energy for our different chemical reactions. NAD, FAD, those are electron carriers that we're going to see soon in catabolic pathways like glycolysis or the Krebs cycle, which we're going to talk about shortly. NADP, that's going to be an electron carrier in photosynthesis. Again, we're going to see this later on. All of these allow energy to be captured, stored, and used safely. For enzymes, the fifth bullet point, enzymes are proteins, usually, but not always, that act as catalysts. They have a substrate that is going to bind to the active site. Sometimes the enzyme has an induced fit where they change shape slightly for best substrate binding. They might require cofactors, which are inorganic ions, or coenzymes, which are organic molecules. And remember, when it's lacking its cofactor or coenzyme, it's called an apoenzyme, which is not active. When the cofactor or coenzyme is bound to the enzyme, we call that a holoenzyme, and that's active. Finally, the last one we just talked about, competitive inhibitors compete with the substrate for the active site, and the effectiveness of the competitive inhibitor depends on its concentration compared to the substrate concentration. They have to be close. For non-competitive inhibitors, we said they bind to the allosteric site, and it changes the shape of the active site. And they work even when they're at low concentration because they're not competing with the substrate. All right, in section 8.2, we're going to look at glycolysis and the Krebs cycle. So this is the breakdown or catabolism of carbohydrates, and we're going to look at how we harvest ATP out of that, as well as generate some high-energy electron carriers like NADH. Carbohydrate catabolism or breakdown is a major energy source. It's going to provide ATP and intermediates for biosynthesis of molecules like proteins, lipids, and others. And because bacteria and humans share many of these pathways, studying them really helps us in the fields of medicine and microbiology and many others, such as antibiotic development. Our first pathway is glycolysis, sometimes also known as the EMP pathway, named after the three scientists who discovered the pathway, Emden, Meyerhoff, and Parnas. So glycolysis is kind of what it sounds like. We're going to break down glucose. And glucose is a 6-carbon compound. This is going to happen in the cytoplasm, so glycolysis happens in the cytoplasm and does not require oxygen. So overall, what you're going to see is that the 6-carbon molecule is going to be broken down to generate 2 3-carbon molecules known as pyruvate. And since overall this is catabolism, we're breaking a larger molecule down into simpler, smaller subunits, it's going to release energy. So at the end of the reaction, we're going to generate a net of 2 ATP through a process known as substrate level phosphorylation. This means you're literally going to pull off a phosphate group from another molecule and attach it to ADP to generate ATP. We're also going to make 2 NADH molecules. These are high energy electron carriers. And then those 2 3-carbon molecules I described earlier, those are called pyruvate molecules. If you look closely at glycolysis, there's actually 2 phases. An energy investment phase, where you actually have to put energy into the reaction at first, and this is going to use up 2 ATP. And then we're going to get our energy payoff phase, and that is going to produce 4 ATP as well as those 2 NADH. The reason we have a net of 2 ATP produced is because even though we make 4, we used up 2. So 4 minus 2 yields that net of 2 ATP. So here's a closer look at glycolysis. We have our energy investment phase. So here we're going to use 2 ATP, and you can see that's going to be used to basically cut glucose in half. There are actually many sub-steps. We're just focusing on the large or big picture. And at the end of the energy investment phase, we've created 2 glyceraldehyde 3-phosphate molecules. So you can kind of see where each of these phosphates went. They are attached to the ends of each of these 3 carbon molecules. Then in the energy payoff phase, we can see that we're going to generate 4 ATP total, as well as 2 NADH molecules, these high energy electron carriers. And I can see that this happened through substrate level phosphorylation, the generation of ATP. And that's when you literally pull off a phosphate group from a molecule and attach it to the ADP, adenosine diphosphate, to make adenosine triphosphate. And that happens there on the right as well as here on the left. So substrate level phosphorylation is a method of producing ATP molecules by removing a phosphate group from an organic molecule and directly transferring that phosphate to an available ADP molecule to produce ATP. So here's the molecule, the organic molecule I'm stealing the phosphate group from, and I'm going to attach it to ADP, adenosine diphosphate, to produce ATP, adenosine triphosphate. And you can see this no longer has that phosphate group on the left. Besides the typical EMP pathway of glycolysis, there are alternative or other pathways similar to glycolysis that exist as well. So some microbes will either use the Entner-Duderoff pathway or the pentose phosphate pathway. And sometimes this is because they don't have the enzymes available to go through the typical EMP pathway. Other times they go through these pathways because they need other precursors. So for instance, in the Entner-Duderoff pathway, some microbes need NADPH for survival in their environment, so they go through this pathway instead. For the pentose phosphate pathway, sometimes microbes will go through this because the intermediates in these pathways or in this particular pathway will make precursors that they need to make nucleotides for DNA, RNA, or amino acids that they need for proteins. Don't worry about the specifics. You can see them in Appendix C if you're interested, but we're not going to memorize the pathway details. So just know that these 2 alternative pathways and other alternative pathways do exist. So organisms might use the Entner-Duderoff pathway or the pentose phosphate pathway if they need other nutrients for growth, such as NADPH. For the pentose phosphate pathway, it also generates intermediates that the microbe can use for nucleotide or amino acid production. And for the Entner-Duderoff pathway, this could be because the microbes lack the enzymes for the typical EMP pathway, or again, they might need intermediates for growth in their particular environment. We'll wrap up the first video here. In the next video, we'll continue carbohydrate catabolism.